All right, so we are on chapter 10, section 6, and we're talking about enzymes. So we talked about proteins in general, and now we're going to really focus on one type of protein, and those are enzymes. So we know that enzymes are typically globular, so relatively spherical proteins, and they're present in every single cell in your body. So the function of enzymes are to act as catalysts to accelerate a reaction. So to make the rate of the reaction increase. And we call that metabolism. So we know because it's an enzyme that the enzyme is not consumed and it's not changed by the reaction. So it'll grab its substrates, it'll induce whatever reaction to occur at a faster rate, then it'll release them and then it can go again, right? So an enzyme cannot make a reaction happen that doesn't normally happen. It just makes the reaction occur at a faster rate. It can't invent something, it just speeds up the process. So a lot of times in enzymes, they're not multi-subunit proteins. And so it's really the tertiary structure of an enzyme that is going to determine its function. So if we look at one example, this is the very first reaction that's going to occur in the process of glycolysis. So glycolysis is basically the first step in converting glucose into ATP energy. And in order to do that, you actually have to invest some energy in order to make it happen. And so the first reaction is really an investment phase where you have this glucose that can be transported into the cell. But in its natural state, right, glucose can come in and glucose can go out. And so if you don't modify that glucose, it'll get pumped right back out. And so the very first thing that you have to do is you have to attach a phosphate group to the glucose. And so once you attach that phosphate group to the glucose, now it's basically committed and it has to go through glycolysis inside of the cell. It can't be shipped out. So it's like the point of no return, right? So whenever we show this reaction, we're going to need our substrates, right? So glucose is a substrate and ATP is a substrate because this is where we are going to get the phosphate group that we're going to attach to the glucose. So we're going to take this very end phosphate and what we want to do is attach it right here on the glucose. And so the enzyme that performs this function that accelerates this process is called hexokinase. So hexokinase will bind to both a glucose and an ATP molecule, and it will speed up the reaction where one of those phosphate groups is transferred from the ATP molecule to the glucose molecule because ATP stands for adenosine. So this is the adenosine triphosphate because you have 1, 2, 3 phosphate groups. So that's what ATP means. Well, when that phosphate, that third phosphate gets transferred to the glucose, now you have adenosine, but you only have 2 phosphates. So they call it ADP, adenosine diphosphate. And so whenever we look at a reaction like this, usually the enzyme name is going to be above the reaction arrow. So that's why we see hexokinase there. And there's a particular portion of the enzyme where our substrates will bind. And we said before that that was the active site, right? That's where catalysis actually occurs. And so a lot of times when we talk about substrates, instead of just saying a reactant, we say a substrate because that means that this reactant actually has to bind to an enzyme in order for it to function. So that's the difference between reactant and substrate. Now, enzymes can function all by themselves, but typically they don't. Actually, they usually need help. And there are 2 different kinds of help that they can get. They can get help from cofactors and they can get help from coenzymes. So if you remember that enzymes themselves are organic, if they need help from smaller organic molecules, those are coenzymes. But if they need help from smaller inorganic substances, think about, right? Organic means carbon, hydrogen. So what if you have an ion that helps, like Mg2+? That's a cofactor. That is something that is inorganic that is going to help our enzyme to work, right? And so enzymes are super insanely specific for their substrates. The active site of hexokinase fits D-glucose and D-glucose only. There are some enzymes that will fit a few substrates, but it's really only like one or two. And they fit very, very tightly, so that only a few things can fit inside of that active site pocket. Think about, nobody else is really going to wear your shoes, right? It fits very specifically your foot, right? So your foot is kind of like the substrate, and the shoe is kind of like the enzyme, right? It's very specific fitting. So there are two general mechanisms, right? So we know that we have an enzyme, and if we add a substrate to that enzyme, we are going to make an enzyme substrate complex. So this is kind of how we write this reaction. And then that enzyme is going to change the substrate into the product, and you will have the enzyme left over, right? So this is kind of our reaction mechanism. And so there are a couple of different models that explain how this substrate, this enzyme substrate intermediate can actually form. And so the two major ones are the lock and key model and the induced fit. And in biology, you've probably heard about the lock and key model. It doesn't exist that much. It's not the typical way that enzymes work. So in the lock and key model, the active site is fixed. The substrate is fixed. They are both rigid and inflexible, but it's like a puzzle piece. It fits in perfectly. It's the exact complement to the substrate, and that's how you get the enzyme substrate complex. But that's not really how lots of molecules work inside of the cell. They're constantly kind of changing and moving and that kind of thing. And so really the more prevalent thing that we've seen is the induced fit model, where the substrate has a particular shape and the enzyme is roughly complementary. So it's really close, but it's not quite. But when that substrate gets into the pocket and starts making interactions, then the enzyme can change and actually take basically the shape around the substrate. And so both the enzyme and the substrate themselves are a little bit flexible. And that's really what we normally see. And so if you look at this example, right, here are our enzyme and our substrate. This is hexokinase and glucose, unbound. So here's our unbound, and here's our bound states. So what do you see? Do you see a rigid, unchanging substrate and enzyme, or do you see something more flexible? And what I see is something more flexible, and that's exactly what happens as that glucose comes into the pocket. The closer and the closer it gets into that pocket, it starts making interactions with the amino acid residues that are in the active site. And that induces conformational changes, so that you kind of have those two globes, the two outer parts here and here, that kind of fold inward and protect and surround the glucose in order for it to be phosphorylated. So this is an example of induced fit model. Okay. All right. 10.6. Yeah. I'm like, which section were we on? So what's the point of an enzyme, right? The point of an enzyme is to increase the rate of a reaction. How does it do it? So now you got to remember the energy diagrams and Dr. B on the bicycle, and that's a really big hill. And that really big hill is the activation energy that I have to get up to in order to go down the hill. So this is the normal activation energy. It's the uncatalyzed reaction. Oh my gosh, Dr. B is going to have to bike up that huge hill. Oh, but if the enzyme bonds to our substrate and starts making those interactions, right, between the enzyme and the substrate, so between the amino acids of the active site and the amino acids and and whatever it is on the substrate, then you can actually lower the activation energy. And so now I have a smaller hill to climb and another smaller hill to climb. Okay, I can do that, right? So that's what an enzyme will really do for us. And the other thing that an enzyme does is that when the enzyme substrate forms, right, you have that hexokinase that has to bind glucose, but it also has to bind ATP. And so if those substrates are not aligned properly, right, if that phosphate group is not close to where it needs to be on the glucose, then they're not really going to react. And so the enzyme puts those substrates into the correct conformation. They're in close proximity. And so they don't have to like just randomly bump into each other like they normally do have to in solution. And so it speeds things up. So here's kind of our summary of how this works, right? Orientation. This is where those substrates have to be in the right orientation. If they're just floating around in solution, well, randomly they might bump into each other, but that's going to happen pretty rarely. But if you have an enzyme that is going to bind to those substrates in a particular conformation, it's going to hold them in an orientation such that here's the change that's going to have to happen, right? And it orients them perfectly. And so that arrangement of the amino acid side chains in the active site will create interactions that orient our substrates. So now that they're oriented properly, now we can lower that activation energy and catalyze the reaction. Okay, so how does this work? Well, when your enzyme is going to interact with the substrate, it's going to form, and I'm going to say intermolecular interactions with our substrate. Well, when that happens, it's going to weaken the bonds that are inside of the substrate molecule. And the book kind of calls them as strained bonds. But because these strained bonds are weaker, then that means that it's easier for them to react with other molecules. And so the weaker bonds will break more easily. And that is what in effect activates our, lowers our activation energy. So in this case, right, here's our enzyme active site, here's our glucose, and our glucose is interacting with, here's a side chain, here's a side chain. These are all side chains in the active site of the enzyme. And those side chains are interacting with different parts of the glucose. And we have something here that is an inorganic thing that is helping the enzyme, right? This is magnesium. So this is a cofactor. So it's required for the enzyme to work because it's going to interact with the phosphates. So remember, phosphate groups are a phosphorus, and then, right, there's another one, but then it's oxygen, oxygen, right, and some double bonds. So what do you have? You have negatively charged, and I guess I should have put it on the other side. So what you have, the negatively charged oxygen there, right? And so that negatively charged oxygen is going to end up being stabilized because it is interacting with the positive charge on the magnesium. And so that is actually weakening this bond right here is weakened. So now this bond will break and we can make a bond between the carbon here and this phosphate. And so what we get is glucose now with the phosphate attached to the glucose molecule instead of attached to the ATP. So now we have ADP. ADP is released, and then glucose 6-phosphate is released. So our glucose with that phosphate group attached. All right, that is it. Let's go answer some questions. All right, so what is the name given to the reactant of an enzyme catalyzed reaction? We call this one a substrate. Substrate. What is the name given to a small, organic, if I could scroll up, organic non-protein part of an enzyme that is involved in catalysis? If it is organic, then it is a coenzyme, right? Because enzymes are typically organic as well. So that's a coenzyme. What if they said inorganic? Come on, inorganic, right? Same thing, but they swap the word organic and inorganic. Like, for example, magnesium 2+. Then this would be a cofactor. So just keep that in mind. You got to know them both. All right, let's see. Oh, I started the wrong ones. That's okay. We're going to do this. What is the name given to the area on an enzyme where catalysis occurs? That is the active site. right? The active site. We'll go ahead and finish this one and then we'll continue to skip like we should be. So what level of protein structure is involved in the formation of an enzyme's active site? So remember we said most enzymes do not have multiple polypeptide chains. And so what ends up happening is it is the tertiary structure that ends up being really, really important in the formation of the active site, right? Because that's all about those side chains and how those side chains can interact with the substrate. Okay, which model for enzyme substrate interaction describes the action of hexakinase? Well, we know that there are two models. There is the lock and key model and there is the induced fit models. So which one do we think is hexakinase? So if you remember hexakinase, I said that when glucose comes in, right, when it starts making interactions inside of the active site, then these globular things kind of like curl inwards. And so that's changing. So what that's telling me is that hexakinase, which model? So we're going to say induced fit model because it's changing. So describe how the enzyme changes when the enzyme and glucose form the enzyme substrate. So the first thing is that glucose has to enter. So when glucose enters the active site of hexakinase, what's going to happen? Well, the enzymes, and they're called lobes, they're called the left and right lobes. Kinase, the left and right lobes, and that's what I kind of circled. This is a lobe and this is a lobe. Will kind of come together and wrap around the glucose. Come together and wrap around glucose, and so that it fits snugly. Excuse me, so that it fits snugly, snug, S-N-U-G, I guess, into the active site, forming the enzyme substrate complex, right? So that's induced fit. That's a definition of induced fit. Okay. So what kind of interaction attracts the cofactor Mg2+ and ATP to one another? Hint, look at the structure of the phosphate group. So if you go back, I got this from chapter 4, section, I don't remember, but it was the one where all of the functional groups were, right? So here's the structure of ATP. So this is our adenosine right here, and then the triphosphate, here's 1, 2, 3 phosphate groups. So if I have a negatively charged oxygen here, if I have an Mg2+, I am going to actually have an ionic interaction that's going to happen, right? So that's a really, really strong interaction, right? This is magnesium 2+. This guy has lost 2 electrons, so he's negatively charged, right? So the negative charges on the phosphate, PHOSP phosphate groups, are attracted to the positively charged magnesium 2+ ion, right? And so because that attraction is so strong, it will weaken the bond between, let's see if I could highlight this, between the phosphate and the oxygen there, right? So it will actually break that bond. So this is an ionic interaction, NTR. And that's very, very strong, right? And so that helps to weaken the bond so that you can then transfer one of these phosphates onto glucose. Okay. And I think that is it. It is. All right, enjoy the rest of your day.